Hello everyone, I'm HEERANYAA NADARAJA. Now I'm going to perform on protein isolation in lab stimulation. Proteins are the biomolecules composed of amino acids. The protein forms the building block of the system and perform most of the biological function of system. What is protein isolation? Protein isolation is a process by which the protein from the cells or tissue are recovered for the analysis purpose. There are several objectives for protein isolation. Protein isolation performed to compare the structure of the protein by different organisms. Protein isolation also performed to purify a protein in order to identify the gene that encodes it and to resolve the protein by STS page, which also known as sodium dodecyl sulfate polyacrylamide gel electrophoresis. The protein isolation also used to examine an enzyme in a crude extract for physiological studies. Now we're going to stimulate. As usual, we have to go to level 3 and let's start. Context. First, in this stimulation, we will learn to purify the protein expressed in E. coli cells from a recombinant plasmid. Here we have several materials which can be used in this experiment, which are reagent, micropipetting equipment, empty tubes, and other equipments. First, there are two E. coli solution, elution buffer, which used to release hydrophobic proteins, lysis buffer, which used to break open cells, binding buffer, to wash out protein that are hydrophilic, wash buffer to wash out protein that are somewhat hydrophilic, column equilibration buffer, which used to separate the chemical compounds, and balancing tube. Here we have some kind of micropipetting equipment such as P2 pipette, P20 pipette, P200 pipette, and P1000 pipette. And these are the tip box for those particular pipettes. And here we have two empty tubes which are RFP tube and super tube. And other equipment such as micro centrifuge to collect the liquid at the bottom of the tube for the easy transfer, biohazardous waste to dispose the hazardous waste resulting from the experiment, including the micropipette tips, waste collection jar, timer, vortex to mix small volumes of the liquid by agitating the tube, and chromatography column to separate the different chemical compounds. The first procedure is pellet the cells. Spin the EC1 tube for 10 minutes in the vortex. In 10 minutes. Oh, microcentrifuge. Open the microcentrifuge and move the EC tube back to the tube rack. Open the microcentrifuge. EC1. Open the EC1 tube. Yes. Open the waste collection jar. Yes, it's already open. Leaked. Okay. Next, resuspend the culture palette in EB. What is EB? Elution buffer. Pick P200. Yes. Set 150 microliters. Oh, save volume. Okay. Open the P200 tip box. Attach. Close the tip box. Open the elution buffer. Yes. Move the micropipette into it. Then move your micropipette into E1. Close the and then close the. Now remove the micropipette over the micro tips. We have to eject the tips and then back to this. Next, move the EC1 tube to the vortex for 10 seconds. And then we have to keep it back. Pick up P200 tip. Yes. Open, P200 tip. You have to attach it. And then close the tip box. Open lysis buffer. Move it over to lysis buffer. Then move it and then open up E1. Put it until the second stop. And then open the hazardous pause. Eject the tube, the tip, and then close the acetus box. Keep it to this place. Okay, then repeat the centrifuge step. Centrifuge step. This is the centrifuge step. You have to keep it into the centrifuge and then wait for 10 minutes. Then close it. Set a timer for 12 hours to make sure all the tubes are closed and incubate E1 tube overnight at the room temperature. We have to keep it into here, into the microcentrifuge. Make sure all the tubes should be closed and then set timer for 12 hours. Start. Okay, next, equilibrate the column. Place the waste collection jar under the valve of the chromatography. Should open and keep under the chromatography. Select the column to drain the liquid inside. Select the column. Yeah, done. Then spin the E1 tube again for 10 minutes. The tube is already inside the microcentrifuge, so no, should start it for 10 minutes. With this, use 200 microliters. Save. Open the two P200 tips attached, close the tip box, and then open the, you have to back it to the rack. Then open the E1. Move it, go over that. No, it should be first stop. Then open the supernal. Over the supernal and dispense the liquid. Open the biohazardous. It should be eject. Close the biohazardous waste and then keep it to the place that we keep. Next, equilibrate the soluble fraction in BB. What is BB? Is binding buffer. Open the P200 tip box. Move the P200 pipette. Attach it. Close the tip box. Open the PP. We should close as this. Open the PP buffer. Move it over here. Should first, then into the super. Open the super tube. Keep it here. Now open the hazardous box. We should eject the tip. Eject it. Now keep it back to the place. Next, apply the soluble fraction to the column. Pick P100 micropipette. Set it as 400 microliters. So it should be 40. See. Open the tip box. Attach the tip. Close the tip box. I should close the tips. Was already the tip. Yeah. Now move the key. Now move the micro to the subpanel. Okay, now move on the top of the chromatographic column. Then eject it. Yeah. Now open the biohazardous waste. Eject the tip. Actually, we ejected already. Yeah. Now place the waste. Now place the waste jar under the wave and then open. Done. Next, wash the column. Set up P100 as 1000 microliters, which is equals to 1 ml. Say volume. Attach the tip. Close the tip box. To draw it to the wash buffer. We should close this. Dispense the solution into the chromatography column. And then open the biohazardous box. Remove the tip. Okay. And then open. Keep it back to the box. Done. Next, using the new tip, drawn 1000 into close it. Now we can wash the tin. Then we have to open the RF tube under the open the column to capture the now close it, place it back. Should check the tip. Should open the That's all. Or actually, predicted results are pretty.